Okay, folks, let's get started. So, welcome to our CNRE Fall Seminar Series. Today's speaker is Professor John Estrada from Mechanical Engineering. John has a very a lab that does very interesting work it's an experimental lab that looks at soft and biomaterials. So John was trained in material science and engineering at MIT, and then has a PhD in solid mechanics from Brown. In his lab, he uses optical methods, to look at materials from the perspective of material and system characterization. He's been in the mechanical engineering department since 2020, and is the recipient of an NSF Career Award for his work. So I'm really looking forward to hearing from him, and he is both sorry and not sorry for works to public as far as his work today is concerned.
Cool, thank you, Mahesh. Appreciate it. All right, so I want to start with where this work came from initially. There's a bit of a history to it. So broadly, my research is in soft and compliant materials, mechanics of materials more broadly. So many of the things I actually work in are, I would say the vast majority of things I work in have a water component to it. Hence, I'm kind of split between these two fields, and part of why I've become involved in the CNRE in general. But where this particular work came from was in understanding injury. Actually, it started as a function of, and this was funded by ONR, mind you, similar to I think this CNRE. It started as, do divers get hurt from blast waves, and how? That really was the initial question that led to where this work started. So, the injury part, where it's arising from is bubbles, essentially, where divers exposed to blast waves underwater due to charges and what have you, suffer brain injuries, and they want to know why this is happening, and it's due to bubble formation, or at least we hypothesize it's due to bubble formation. Bubbles that rapidly expand, collapse, and oscillate, which we know as inertial cavitation. All right, so the question that we wanted to answer, though, is how much pressure, how much damage corresponds to how much injury? It's a mechanics question fundamentally, right? Of, something happens, an external load applied happens, right? And then how does that result in human being injury? And like I said, the mechanism here is bubble expansion and collapse and oscillation. This expansion, collapse, and oscillation produces kinematic effects on the environment, right? You have a bubble that expands, it pushes material out of the way, and the result therein are stresses that will serve to damage the cells inside this brain. The way it happens is an energetic dumping process. A bunch of energy goes into one location, it causes this rapid motion. Now, in principle, it doesn't have to just be ultrasound or extremely fast blast waves. Really, this could be any kind of energy deposition. So we use, when we actually study this in the lab, we use laser-induced cavitation as a reasonable proxy for the effects of focused rapid waves. But the way we proceeded to analyze this was we start with a biological sample. We then impose some kind of mechanics on it via dumping in a bunch of energy. We create this dynamic process, which is expansion of a bubble. It collapses, oscillates, does its thing, and then we see resulting damage. And we want to know how much bubble size corresponds to how much damage. So we do this, like I said, I'm an experimentalist. So we did this in the lab to actually generate data. Let's see what we actually measure. And we send a pulse laser in. That's the speed of light right there. Don't worry about it. You're very fast at seeing right now because you're eating shawarma. That's what lets you do it. So anyway, the laser goes in up through the microscope. We then measure that at really fast rates, order of 500,000 frames per second plus. And we generate cavitation at a focal point, which then, if this video will play, expansion, collapse, oscillation. This is 500,000 frames per second we're looking at. All right. So if we show this as a time series, again, it's a mechanical process. On the left side, we have a pristine biological material. As we go, this expansion happens. It pushes things out of the way and exerts hoop strain on the material, right? As well as radial compression, resulting in eventual damage that we want to measure. And we do this by looking at actual image stacks of our material. But this is our mechanical event. We have a radius expanding that goes to some maximum, collapses, oscillates, and we end in some kind of quasi-equilibrium with a bubble there, where it's really just the water and some of the organic stuff that's vaporized and become a vapor locally that sticks around for a little period of time after. But this kinematics that we measure right here is what defines our mechanical event. Then we just have to cross-check this with what kind of damage we had, and it looks like this. So this is my brain. No, this is not my brain. This is rat neurons that are in a gel. They're collapsed onto a 3D stack. And this is where the bubble happened, where we put it in experimentally. And you could kind of see over here, these guys look sort of normal. And those of you on Zoom, I'm pointing over on the left side of this green image. Over there, low stretches, farther away from this bubble expansion. We have relatively low amounts of damage. As we continue to move this way closer to the epicenter, where our stretches locally were larger, we had more damage. In fact, in this particular one up here, I turned it on its side, there's like a region where, and I'll kind of highlight this on Zoom too, right here, we have pretty much all of the arms get ripped off kind of process, where there's just a critical strain that broke a whole bunch of stuff. And we want to figure out what that exactly is. But the important finding here is that the damage scales with bubble size also. And this was after running hundreds of experiments here. I was a very active PhD student. But further questions remain, right? This isn't the be-all, end-all. What if we had mechanical gradients in our material? Brain tissue is not just uniform. We have white matter, gray matter, anisotropy, folds in the brain. All of these could play different roles. Do we need other physics from our history for models? How could we reliably, even characterize gradients if we had them, or other physics if we had them at larger scales? And finally, if we take a completely different system but want to use the physics of cavitation to do something positive, how would we do that? I'll mention that like my group is built on some of these. I have active projects going on in a whole bunch of these different areas. They're all over the place. I have, folks at other institutions we're working with on some of these. But the one I'm going to focus on today is an all-Michigan study. This is using bubbles for drug delivery. And we're going to discuss how we can actually harness them, and hence why I'm not sorry to burst the bubbles as opposed to sorry previously. All right, so we intentionally want to use bubbles, which means we're going to start with a matrix that contains droplets in it. These droplets are going to contain drugs, and we want to use the drugs to promote something called angiogenesis, which is essentially the formation of blood vessels. Angiogenesis is a tricky thing. Folks with diabetes, for example, have trouble making new blood vessels in, for example, if we want to replace tissue at a diabetic ulcer site. Those folks with diabetes often have amputations due to just being unable to heal from those kinds of processes. So we want to put in actual drugs that will encourage blood vessel formation. But it's a tricky process because you actually need a drug that'll start it with all the precursor stuff that has to be kind of gently treated so you don't break it all up before it gets built. You don't want to destroy all the Legos before you build the tower. But you also need to stop it at the right point. Otherwise, you over-proliferate blood vessels and you actually destroy the tissue also. So there's like a Goldilocks region. So we need droplets that contain two different types of drugs, or one kind of droplet containing one and another kind of droplet containing a different drug. More on that later. These droplets are commonly used in radiology applications, and hence my collaborators are in the radiology department. Mario Fabielli and Mitra Aliabuzar, who are absolutely stellar research scientists, are the ones that really are just superstars on this project. Now, these droplets have an elastic exterior just to kind of hold them together, but they are essentially perfluorocarbon, it's like an organic liquid droplets. And they contain a drug in them. They're emulsified, more or less. But we actually have a choice of what kind of core we want to use, and those choices have different lengths of perfluorocarbon, meaning that they actually have different boiling points, different resulting necessary pressures to make them actually pop when we want to go ahead and do that. And so the smaller the chain, the more volatile it's going to be, más o menos. All right, so from here, we have an experimental setup that we can induce the popping of these bubbles. We can phase change them, trigger that phase change. And what it looks like is we have an ultrasound transducer that's pointed at the sample. We send in a pulse, the pulse will pop the particles, more or less. All right, but we have some choices around that. We can pick what frequency, how many pulses, like a whole bunch of different things, as well as even what the matrix is made out of, which is where my group really comes in. And it looks like this. The procedure, we put in this pulse, we have a particle that's originally here, and it turns into a gas afterwards. And this green will diffuse into the environment around here, which is a drug being delivered into its surroundings. We want to have enough drug to create angiogenesis and enough of these particles to release enough drug. But remember what happened with the cells in the neuron project. If we expand this too rapidly, what happens? We break all this stuff, right? We don't want to break our material or our components, so we have to be gentle about it. And this is the art form that we're going to go into. So how this is happening, actually, it wasn't known. They didn't know these were inertially cavitating. We just knew they were popping. And so what we found initially was that this had an inertial component to it, meaning essentially that you have to take the acceleration piece into account, right? Inertial just means you have your mass and acceleration term in your equation of balance, right? Your balance of momentum. And so we had to take that one into account. You can create cavitation quasi-statically too. If you just load an environment or pressurize an environment locally, you can cause a material to rapidly expand. But only once you go past a certain speed of injection does this inertia start becoming really important. And the resulting deformations also get extremely fast. Like quasi-static cavitation happens on strain rates of like, sub 1 per second. The inertial stuff, we're talking 10 to the 3 and up, depending on your length scale. Smaller length scales have even higher speeds. So, this isn't something that we don't know anything about. Cavitation's been actually harnessed usefully in therapeutic procedures. You may know it as the thing that destroys pump impellers, where you spin a pump impeller at high angular rates. The result is eventually you get erosion. And it's because of these bubbles actually collapsing and hitting the surface of this impeller at the trailing edge really, really hard and quickly. And quickly. But you can also use the same principle to do positive medical things, like you can fragment tumors, which is there was a recent article and also a new technique or a new clinical technique developed at Michigan called histotripsy. If you saw these bubbles break or these bubbles kill cancer anywhere on campus, that's what it's about. There's a group in the BME department that uses bubbles to break up tissues, led by Jen Shu and others over in BME. You can also use it to break kidney stones up. If you put a bubble over and focus it at a kidney stone, you can slingshot it and break up the stone, which is really useful. So if we're doing it effectively and control the physics, we can actually leverage it for something. And that's what we really want. But we need to know other things about our behavior, for example, the material properties, in order to actually do something positive with it, as opposed to just let it run and see what happens. All right. And we kind of learned something about this, at least from this first study, which was that there's a length scale dependence. And now I want to point to like kind of the numbers here, right? So there's a stretch ratio dependence on where these things are damaged. And because of that, and also that we saw cavitation was really the thing that damaged the cells and not just putting in laser energy. If we put in laser energy and we don't have bubble deformation, pretty much nothing happened. They were completely fine. But there's this difference in injury depending on how far we are away, and cells closer to this epicenter were more damaged. What we noticed, though, is that it's actual bubble size dependent. It's an energy dependent process. And we wanted to really focus in on that. And so it's not just that a certain radius is damaging, it's that a certain distance away with respect to the radius is the problem. So it's actually a strain dependent criterion. So we have like a failure strain kind of material property here that we actually need to work for. And I showed these before, so I'll just kind of keep on going here, but that's kind of what we've got. So now, putting this in context of our current problem, we want to do essentially two things. We want to use this injury framework to help us understand the mechanics in these scaffolds. And we need to do so with the context of material behavior, viscoelasticity of our surrounding material. And this is going to help us hopefully distinguish between our types of particles so we can inject, or not inject, but permit one drug to be used to create blood vessel formation, and then stop another one later with a different kind of energy signature, all the while never breaking the stuff surrounding cells or biological components when we trigger the phase change, right? So we have to initiate two different particles without breaking stuff in the environment. Kind of a Jenga problem, right? We don't want to do damage. We don't knock the tower down, but we do want to actually pull out the things that we want of interest, right? That's what we want to do. And then we also want to characterize, experimentally, did we do it or did we not? And that's with cell damage mechanics. All right, so we'll start with part A. And we have a procedure in my group for doing this. We can understand the mechanical interaction between bubbles and their surroundings using both experiments and theory. We connect them together and we get characterization, essentially. So I've shown the experiments, we put a laser in, we create these bubbles and watch their dynamics go. We compare this to a theoretical numerical model. You guys probably have done similar things in classes with a freely oscillating bubble. You may have seen the Rayleigh-Placet equation or heard of the Rayleigh-Placet equation, perhaps. Yes, no, maybe, yes. Rayleigh-Placet equation comes from balance of momentum. You have the assumption of incompressibility that goes in there, and you have it in spherical coordinates. You integrate it from R, your material over your material, and you get some function that has an inertial term in it. So that's what we're using with a bit of extra physics. We have finite wave transfer we need to take into account. We have heat and mass transfer that are important in the physics of this problem. So they're just, a couple more bells and whistles, but the fundamental stuff is the same. It's kind of what you've seen before. And we go ahead and actually forward simulate this with some guesses of material properties. I'm realizing I have an X on my arm, which was from my statics class this morning. Sorry if this is distracting. It's not actually a tattoo. I just drew on my arm with marker this morning in class. Great. So we combine these two, the experiments and the theory together, and we go ahead and iterate in mechanical properties. We alter the mechanical properties. You can use an algorithm to do this to kind of get to a best match, but we have some cost function space that we want to try and say, if we pull out the radii from the simulations with the right material properties, they'll agree perfectly with experiments. That's what we're hoping for. That's where this X is showing it's the right material properties. And we can go ahead and say, okay, this is what our material behaves like. So we've used this before, and I'll just describe the model a bit more because some of you may have some plasma interest here. I don't do that. So I'm going to say right now, I avoid all of the plasma stuff and say model begins at the top where we have all the recombination, everything is kind of done with. And we start with a bubble at maximum size in a material, and we're going to let it play out, and it will come to some equilibrium later. And, we measure these, we test soft materials here, and, we kind of have shown that this works in the past and can characterize different materials this way. All right, so I want to show you how it really plays out intuitively. And so if we have an experiment like this, and we go and say, what if the material were completely inviscid? Let's say it's just a fluid with very low viscosity. The results from the simulation, assuming the bubble maximum minimum, or bubble max and equilibrium were kind of like this is what the prediction would be. This is like an inviscid fluid. So if we increase the viscosity in the simulation and run it forward, as we go from viscosities, of 1e minus 3 Pascal seconds of water to, water, milk, kerosene, flax oil, all the standard beverage offerings at Pierpont. As we go up in nice warm glass of motor oil, as we go up in viscosity, the envelope drops. Right, we have a suppression of this rebound that can happen because we're actually sapping energy from the system. So this envelope drops, and the time that collapse actually happens moves out. It gets delayed a little bit because some of those dissipated mechanisms will restrict that from happening quickly, right? Now, in contrast, if we add elasticity to the system, if we increase the elasticity of the material, and this is using only a pure elastic model here, if we go from brain to sort of conventional soft materials, also offered at Pierpont, as we increase the moduli here, we can actually sharpen the collapse time because you're building an elasticity to the system, which rapidly sends the energy back down. So that kind of gives us a trade-off that we can look at even in just that one parameter of when collapse happens. And we've started using that as like a very quick rheometer technique. All right, so generally, takeaways, stiffness speeds up collapse, viscosity slows it down in the way the system plays out. And what we want to do is actually combine that into kind of like a reduced order model, more of a parsimonious model, but like, we want to say if we slow down collapse one way and speed it up another way, maybe we could use that balance to tell us what the material behavior is. And we do. So it turns out they have this kind of inverse square relationship on the resulting collapse time. And not only this, the really useful thing here is that the viscous behavior is actually size dependent and the elastic one is not. So the elastic speed up normalized to size is always the same. But for smaller bubbles, you have more of a viscous effect, similar to how for smaller things, you have lower Reynolds numbers, right? It's the same principle. It's also like a Reynolds number, essentially. So when we go smaller, if we have experiments of different sizes and the material's the same, we might have a lower Reynolds number, but our viscosity stays the same. And we can use that change in sizes to really get us characterization here. So go ahead, yeah.
The period is the same to maintain, or is it getting shorter? Like touch in your dam isolation. Yeah. So the periodicity actually.
Everything, yeah, everything is shorter, actually. Yeah, the periodicity gets shorter. Yeah. With time. This is real time.
So you have like a transform.
Yeah, we could. Yeah, right. So there is for small deformations. Like we could get the frequency of the system with elasticity included in a small deformation regime. When we go to finite deformations, it becomes harder because the initial collapse actually like the widths of each of these peaks, these are kind of consistent, but over here they're different. And that difference is enough to screw up the transform.
Well, so how do you know when you transition into the bunch of
Good question. Good question. I mean, it's so large deformations versus the linear regime is generally how different percentage-wise you are in some quantity from what you see in linear. So it depends on the quantity, right? That we care about. But
do you worry about the heterogeneity of the medium happening, or is not a factor?
that's a different factor, right? The heterogeneity of the medium will produce other effects which are going to be size scale dependent largely. So yeah, that's also its own challenge in the system that we'd have to care about more at the maximum expansion when you have a larger length scale, because your strain field feels more of what could be a heterogeneous environment as opposed to the small scale, which might be more of a locally homogeneous environment.
Yeah, I'm worried, not worried, I guess is not the word, but I wonder, I suppose, you have your mechanical wave propagation,
Sure.
essentially, and then if you have other E&M radiation,
Sure.
for example, you have a scattering medium, then that can sometimes look like you have or exacerbate the absorption effects. Right?
Sure.
Damping effects see here from the model could be exacerbated by scattering.
By scattering, okay. Sure. Actually, that would be, if you have like time to chat about that, I'd love to hear more about that if that's cool. Just because I hadn't thought about. I mean, we've looked at heat and mass transfer for sure, but E&M specific scattering we haven't considered. So that would be like of interest. If
like whatever loss component we talked here will be exacerbated because
right.
there's
Right, right. And so actually maybe this gets to even like a better point of why we may want to not use these whole curves and instead use as low of the as little of this dynamics process as we can. And for that reason, we kind of like this collapse time as a metric actually, because it's just done at collapse. And really like we can treat the energy, and maybe I'll just kind of get into my next point here. We can get into the energy as like an energy balance problem where we have like the work done kind of by the bubble, and then with maybe this is just the viscous effect here, and we could add more viscous terms for other kind of dissipative mechanisms, right? But in principle, it's some kind of effective viscous behavior. And at least so far, like our experimental work and rheometry work in different oscillation scales have been pretty good in agreement. So we think it's still dominated by the the viscous component. But like at least that lets us stay in this like just this one quantity that moves, as opposed to worry too much about what other kinds of dissipation we have later. But it's still good to think about because always we want to get it right. You know, at the end of the day, we want to make sure we're getting it right. Go ahead, Mahesh.
Surface tension look like in this figure?
Yeah, surface tension. So for water, it's just surface tension of water. We do include it. We do include the surface tension of water in this. So this would also be its own pressure-like term, like 2 gamma over R. We do have that in there in our full model. It's actually pretty small in the problem. It's not super high, particularly because like our so we use like a Weber number approach to it. We non-dimensionalize it, and it's on the order of like 2% of the same like other pressure component, the stress-based component. So it is kind of small, but it's non-negligible. So we do keep it around. Yeah. Right. So anyway, long story short, just to kind of keep us going here, long story short, we can use approximated expressions using those collapse forward and backward times to actually fit multiple we can kind of get estimates of what our collapse times ought to be for different choices of material properties. And this allows us to like do this calibration with basically no need for doing forward simulations anymore, at least within the assumptions that we've made. And so it ends up being that there's kind of, you could either make this multiplicative or an additive in small collapse time differences, but there's an effect that essentially adds up in some way of the elastic and viscous shifts. So, you know, what we're seeing kind of like over our like moduli we care about in our group and the viscosity we kind of care about in our group, the agreement between our like best fit simulations and this like reduced order version are really good. They're like within a few percent. And this is well within the tolerance of experimental calibration generally. So we're pretty happy with this method. So let's let me skip to just the experimental data here, just to bear this out. So if we do this, as I mentioned, at multiple size scales, there's a bigger effect of viscosity for smaller bubble sizes. And so our dynamics, even for the same material, if we just have a smaller scale experiment, we get the suppression. And by having multiple different experiments calibrated kind of simultaneously, we can see how this works with our best fit modulus and viscosity, and get this like this I guess difference in our predicted and actual collapse times that we measure using like a double strobe technique in our mechanics. We can construct a cost function out of this and say with pretty good confidence that we know what our material behavior is. All right, so then the next piece of this is trying to figure out what happens when we have droplets in there, right? Because so far all I've shown you is just laser and water, or laser and material containing water. When we put in droplets, it turns out the collapse times don't really change much, which is interesting. So that presence of a perfluorocarbon being in the droplet, and when we shoot a laser at it, doesn't really affect the collapse time very much. It might affect later behavior, and it certainly does. When we have a more volatile gas, it will want to take up a larger volume. But initially, that collapse dynamics does not get affected, telling us that droplets, they don't affect this collapse significantly, and therefore, because that time of collapse is really strongly correlated with their material behavior, this tells us that the dynamics are mostly material, but functions of the material properties of the surroundings, as opposed to what we use for the droplet specifically, which is useful because it then says if we pick a certain, let's say, fibrin concentration for a scaffold for something that's going to go on someone, we don't have to worry too much about which droplets we're going to put in also to be the drug launcher. It's going to be mostly dependent on just what the formulation of that scaffold is. Super useful clinically, right? And so this was a pretty useful finding for us. But eventually more gas is created because we have a more volatile species. And so the later development, the kind of slower order of second scale dynamics are going to be expansive. Also very helpful though, because let's say we want to push the drug into the surroundings more, just use something more volatile, right? So the material properties are what we really care about at the beginning, right? Of do we harm the stuff? Material property dependent broadly. The second piece of do we push drugs into the environment are droplet dependent. All right. So we've now delineated our two effects, which is, like I said, super helpful clinically. All right. And so I'll skip this one and then kind of, let's see, we'll talk about this one now. So what if we were to instead of laser, we'll use acoustic energy. All right, so we're not going to fire a laser in here. Let's try and use acoustic energy instead. So we go ahead and hit this with an ultrasound transducer, which is complicated because we don't really focus on one specific particle anymore. It's kind of a region, and so there can be some combination effects. But as we do this, we have this result of multiple particles showing up at the same time, right? We have cells or these are fibroblasts, but we can have any kind of precursor materials, whatever we need for angiogenesis in there. And we hit these blue particles here with ultrasound, and they're going to start expanding in their surroundings. Now, this is just what happens later, because these are order of seconds. So we don't actually see in this set of images, at least, what happened to the local surroundings. But we know the material behavior. We know the material properties. So we can actually kind of estimate what happened based on knowing that, at least. Now, what we can do is then see how we can actually put measurements on cells and say, not measurements, but measurable tags in cells that will allow us to see whether they're injured or not. And we can get a dependence of the cell injury on, for example, the type of perfluorocarbon we use. And so we've tried a few different ones of these. We tried kind of the internal metabolic activity. We've tried an uptake of a marker called propydium iodide, which ordinarily doesn't get into cells unless they're damaged. They're kind of different ways of doing this. But we kind of, at least from here, can then say, well, if we use one of these fluorescent things as a metric, we can choose something, and then so let's say we use this green stain as calcium, just like we did in that neuron study. By doing that and seeing how close cells were to these bubbles and knowing the material behavior, now we can get estimates of the kind of strain it actually takes to do damage to these cells, giving us something that's now clinically relevant of, let's say we want to know how many droplets we can put in if we cannot break more than 1% of the cells in our volume, right? Because if we have some density, we can know, using compressibility assumptions, we can know as we've expanded these bubbles, how big they're going to get to, because they're all the same size going in, and then how much of that volume is going to experience a critical damage strain. So knowing this threshold takes 2% strain for these two different particles, and then 4% strain for this other one. Knowing this now allows us to back calculate, okay, what's the maximum amount of particles we can put into a volume? So this gives us a seeding density estimate. It's material property dependent also. There's a little bit of coupling with the octane, which we're studying right now. But, you know, this now says it gives us an actual, like, how much of our volume is affected at critical strain. We have a number on it. We can design for it and move forward. And it also tells us that we can actually seed those octane particles because they only damage cells at a higher threshold. We can seed them at a higher density, right? So we can get more drug into our system. That's really critical, right? If you want to make sure it works, you put in extra drug to make sure it works. So we're now going to look at this as far as like different ultrasound parameters, amplitude, for example, frequency, for example. And, you know, we're going to continue getting there, I suppose. But yeah, that's kind of all I had for today. We have other things going on, asphericity, full field methods using MRI. So if you're interested in any of that, want to chat with me about any of those, even informally as a student, by all means, I'm encouraging of that. And so yeah, thank you for having me. Appreciate being here and happy, very much happy to take questions about what I presented. Cheers.
Let's ignore that you're trying to be a medium, but what if you have a homogeneous medium but have different boundary conditions with the domain it's targeting? Like, have you, or are you planning to characterize the excitation and then the effect?
Sure. So the question is about the keeping the medium the same, but changing the boundary conditions of that medium, whether it plays an important role in the dynamics or maybe the behavior of the system, perhaps. This is a great question. And so a colleague and I actually have, we're in the middle of writing a proposal to ONR right now about exactly that. It's using the dynamics in thinner film environments. Because for one, that will result in waves rattling around a whole lot, right? So there's acoustic effects that are very important. The way it can be approached from a modeling standpoint is to have kind of a symmetry. You can use the planes as symmetry conditions and run two bubbles at the same time. And as a result, they get kind of egg-shaped, which is interesting. But it also results in weird aspherical behavior that it necessitates stepping into finite element or like full field flow solver conditions. Now, the compounding of material behavior then becomes its own different kind of important because you can have multiple rates in the same problem, kind of. And so characterizing those rates, if they have like a spectrum to the viscoelasticity, becomes also really important. So I guess the short version to your question is, yeah, a whole lot of stuff gets a lot more complicated, but at the end of the day, a lot of our direct simulation tools are really useful in solving those problems and permit us to like approach them in at least procedural ways, if not invertible, right? So yeah, that's kind of the way we're looking at tackling it, but boundary conditions are critical. They're critical, yeah. Go ahead, Mahesh, and then I'll take it. Yeah, go ahead. By all means. Yes, I am.
Do you think that like energy weapons like lasers, for example, can induce, like, can this be used subversively to induce neural damage?
So I'm going to be careful in answering this question. So first off, the question started with Havana syndrome, which is Havana syndrome essentially was that folks in Cuba, in the Cuban embassy or the American embassy in Cuba were feeling sick after, basically neural sick after being posted there. And then the second part of the question was how it relates to whether someone could be injured by having particles that were seeded in the brain and then focused with laser. So I'll mention this from a different, or describe this from a different standpoint, which is generally it is hard to injure the brain in some sense by getting things into it. And one way of putting this is there's a considerable amount of research being done on what's called the blood-brain barrier, which is essentially the meninges and all the area around the brain creates like a seal that does not allow transfer easily. And cavitation is being used to try and porate it, but only briefly. It's actually quite difficult to do. There's also a study being done here about trying to deliver waves into the head directly and do so to disrupt tumors that, like a glioblastoma, for example, type of tumor that folks want to get rid of. And it's hard because there's major aberration that you need to do this like big aberration correction to get around the acoustic impedance mismatch between the skull and the brain. So it's difficult to get energy even in a focused way into the head. Actually, it's quite difficult. The difference is when you have an actual blast charge, which just has a ton of energy, right? And if it's someone underwater, they're getting the full blast without any air dissipative from like acoustic impedance mismatch effects. So like therein is a pretty major energy transfer mechanism, but even like a remote directed energy weapon that's like, you know, laser like, right? Even if it's microwave or something, it's just hard to focus that energy to the point where it's disruptive, actually. So I would say that's less of a concern. Not that it's not a concern, because I can't speak to that fully, but the scales of energy are quite different once you have the mismatch occur. Go ahead.
Are there simulations or studies that have done the couple problem, including damage? You know, so the first
Yeah,
thing that had the network.
right. That is a so do you mean for the neural study? Like
Yeah, the intro.
yeah. So the question is about have there been coupled studies with neurons and they're trying to do like a combination simulation of the deformation of the material and also the deformation of neurons inside the material, looking for, let's say, connectivity. Embellishing the question a little bit, Mahesh, sorry. No, there have not. So this is for one, the neurons are very squishy compared to their surroundings. So you don't really need to model them separately, I would say, is a big part of it. There have been these like connectivity like studies. There's a guy at Penn State, I think, Ruben Kraft, I think is his name. And he works in he's done work in what's called like a connectome model of you have axons in a brain, you put loading on it, and if you exceed a threshold, then you would get connectivity disruption. That's the closest I've seen, I think, to that. And it's a reasonably good proxy, I think, for what that would be. But no, there's still actually like hot research in brain mechanics of like how to model axons or like extension to couple the mechanics, even in like blunt injury, to damage in the brain. It has not yet evolved into blast-like injury at these really fast rates.
The related question is there work on, you know, taking a unit cell approach, going from microscale to the continuum properties that you're inferring?
Right.
Is there work like that?
There's interest in that. So going the follow-up question to this is there interest in going from like your local, you have neurons in a material, to then a represented volume element type approach in simulations? There's certainly interest in it, and I think folks have been kind of going that direction to assume like an anisotropy of brain matter in different locations for simulation purposes. But the continuum mechanics gets very complicated very quickly with the brain, because you have anisotropic materials connected to somewhat isotropic materials to a fluid, which often isn't actually treated like a fluid in the FEA, classic, you know, abacus-like problem. So there's that, which is difficult. Then there's like all the geometry, which comes from MRI scans, which then have their own error associated with them, which is complicated. So I think, you know, there is certainly in the health and human protection division, code 34 of ONR, like they care about this in a major way because they're trying to build up the digital twin model of people. They want to make sure that like they can get a full representation of a person that would go into simulations, so they can try and make bespoke protective armor or whatever, right? Or know if they're getting hurt while in the field. And it's just still kind of in development, I think, is the you know, they're ongoingly looking for projects doing this for sure. Yeah.
So you showed the really cool cost function to try to derive the stiffness properties and the viscous properties of that material. So my question was, what material was that done for?
Sure.
And are there other like biologically relevant materials that are still an open question, what the G and V?
Oh yeah, for sure. So the question is, what material is in this cost function plot? And are we still trying to characterize other things? So this particular material is polyacrylamide. It's a pretty simple mechanical material. It is, you know, acrylamide is a standard chain. The mechanics are roughly not roughly, they're quite well described by neo-Hookean models. So they're not extremely complicated for this kind of analysis. Now, what we care about, actually, I mentioned I was writing a proposal right now. I mean, we're trying to do completely weird tissues, especially liver, kidney, and muscle, which are all of major interest for blast injury. So, you know, we go from something that's pretty straightforwardly described by a neo-Hookean potential, and we are going to have to step out of that ASAP, right? And so you have to bring in nonlinear elasticity, potentially nonlinear viscosity in and of itself. So we wouldn't necessarily just be able to use a Newtonian parameter in there for viscosity. Being blood filled, it's, you know, that's its own kind of fluid, right? So we'd start with simpler models and build up the complexity as we see we need it, but those will come out of, we'll have to be clever in the way we take measurements, right? And it may be that we have to take different orientations of tissue when we go and do these simulation, we do these experiments rather, as part of that. So yeah, there's a whole lot of characterization that we still need to do, even for like general stuff, but we're building up a library of a whole bunch of different soft materials presently. And it's not just me, it's like there are 3 other groups that we're collaborating with to do this and span that space. Yeah, go ahead.
Picture of the other center. Yeah, this one. I'm wondering, obviously there's some randomness to the distribution.
Yeah.
Axons. But post blast.
Yeah.
There's some damage with the
Right.
axons. Have you done, again, back to this idea of Fourier transform?
Yeah.
Have you done a Fourier transform of the image to see with the symmetry of the damage actually looks like?
Yeah, this is a really interesting question, and I think so. Let me phrase it for those on Zoom. It's kind of how we looked at the orientation via Fourier transform of the neurons in this kind of structure to see if there's a preference maybe to damage along, let's say, a specific direction, like a hoop direction, as opposed to radially, something like that. So there's a challenge here in that this is coming from 3D images. I've compressed them in another direction. I have run a Fourier transform, and it's actually pretty isotropic, interestingly. It's a good thing because we made the cultures by randomly seeding neurons, and they randomly form connections to their neighbors. So the transforms actually do generally give you something like, you know, the nearest neighbor like plots, right? Where they're kind of just rings of your seeding density kind of randomly oriented.
And this is, how big is the sample?
Yeah, these samples are on the order of 1 millimeter thick, and they are on the order of 15 millimeters wide. And the bubbles are on the order of like 100 plus microns. Yeah, good question. So they're certainly lower density than actual neurons in tissue. So the way they're fabricated is we have to take neurons out of rats and like put in a certain seeding density that will allow them to grow, but not let them get hypoxic, meaning they don't have enough nutrients. They can't get enough nutrients because we don't have any vasculature to deliver nutrients. So they have to be lower density than actual tissue. Right. So the seeding density, I forget the number, but it's thousands per millimeter cubed, I think.
Surprised? We're not surprised. The damage, there's no symmetry.
Right.
It's too much symmetry.
Yeah. Am I surprised or not surprised that there's this kind of damage? So I'm unsurprised. The reason being that the kind of failure that we expect is not specifically orientation dependent, it's deviatoric strain dependent. So it's almost like how in plasticity, you have just like a volumetric change in one direction, and then the other volumes get like correspondingly changed in the second and third directions, resulting in just a net like strain magnitude more than with an orientation. The strain in the radius direction is twice as much as in the circumferential. Yeah. And so you could kind of see that as then also the hoop stress effect would be kind of counteracted, because it ordinarily would be twice as much, right, as the radial. So I'm kind of unsurprised that they're not really giving a preferential there, but that's with some caveats. I mean, it also requires the cells to be pretty well encapsulated by their environments. I didn't show that, but they are. There's collagen running all throughout these cells, trapping them in place. So they're really fixed in their environment, and then the environment squishes around them. So that is part of that I didn't show, which is part of why it's kind of expected.
Yeah, it's other types of tissues, the degree of fibrosis.
Right, where it's aligned, right, where it might be aligned. Yeah, fabulous question. I mean, if we have like scar tissue, right, fibrosis, we would absolutely expect some weird behavior, trying to split fibers or pull them apart. And at least in some muscle tissue we've done so far, we do see the deformation going kind of egg-shaped first, owing to the stronger elastic, the higher elastic stiffness in the fiber direction compared to the transverse. So that's unsurprising, but then what the critical strains might be may also be different in along fiber versus transverse to fiber, and we would have to decouple that for sure.
Like close in parts, there's massive portions of cardiac tissue, for example, get fibrotic.
Right.
Can't repair itself actually, in part because it gets more stiff.
Right.
You go in and over time disrupt a little bit.
Yeah. Break it up a little, and then yeah.
There are some, presumably there are some stem cells present in the tissue, very, very low concentrations, but there's nothing that they can do because the matrix is so stiff.
Right.
They can't regenerate because the matrix is so stiff.
Yeah, that's a cool application idea. That's a really cool application idea. Like try and yeah. Nice. Great. Yeah, sounds good. Like let's talk after. This sounds great. Cool. All right. Yeah.
Question is on one of those. Is there any significance to the number? It looks like there's a core, and then there are branches that come.
Yeah.
And it's just like looks like there's something like between 5 to 10, maybe 6.
Sure.
Is there any reason for the number that comes?
Yeah. Yeah, there is actually. So there actually are different cells here. They're not all neurons. In fact, in the brain, there are, and this is actually a disputed quantity too. There are some amount of neurons and some amount of astrocytes. What they do roughly is neurons are the things that signal to one another. Astrocytes kind of grab onto neurons and help them clear out their environment. More or less, it's overly reductive. If there are any BME colleagues in here, I absolutely apologize for that. But more or less, that's what happens. So astrocytes have a ton of projections. They're literally called star cells because they're like, they have a lot of these projections. And so if I were to look at some of them, I would maybe say that like, that's probably an astrocyte, that's an astrocyte, and then one like this with this like really thick projection, this is probably an axon of a neuron. So like there's more at play than just like why they would have some. It's really what they've differentiated into, and their environment as they've grown does create a number of projections that they want. There's a second part to that, which is if neurons reach out to project to something and they find that they can't find anything, they'll just kind of like the arm will kind of go away. And it's because they want to maintain like energetic optimal. They don't want to be sending nutrients to something that's not going to be used. So they kind of figure themselves out also. And it's therefore also a function of like how many neighbors they've had, so they can like reach out and actually get them. So it's a cool system in that, like, yeah, they signal to one another, and that's what keeps them around. Cool. Well, thanks again. I appreciate the time, and I'm, please hit me back to you. Oh, I had this off the whole time. I had this off the whole time. Actually, probably for the better.
This was on, wasn't it?
Yeah, this has been recording the whole time.
Very good.
Yeah.
So you can
Audio's on.
stop sharing.
Okay.
That is very good.
Thanks, guys.
Very interesting.